Hello dear students, very welcome in the very first morning. I hope so all of you doing well and great. And today in very first morning we will go through the solving of biology first paper assignment. In the Zoom, how many students are joining here? Okay, so vice versa, Zoom and live is go on there. I think you saw your 11th week assignment, biology first paper on them, and there is the three part, though I said the two device on there, the assignment on them, so I got that point. First one, bivalent creating a stage, to explain up with some features, and then after crossing over its mechanism and economic and also importance on them, importance. And third one, last two stage of Prophase 1 to compare, make it a comparison on them. In the Zoom who are joining in here, I will just up to see on them the assignment also, vice versa. Sometime you will be hang for a couple of seconds in them, since I need to check out the assignment on them. Here I have seen that bivalent creating stage on them. We will explain about with the features, crossing over, mechanism and importance, and Prophase 1 last two stage difference in them. We will go through on them. And your assignment title, I see the assignment here in given. Assignment title will be the different stage of Prophase 1, discussion of different stage of meiosis, Prophase 1. This is the assignment title. So what will you write in the assignment title in very first time? Title, I will write here the title. Discussion of, you will write up discussion of different stage of Prophase 1 of meiosis. Prophase 1 of meiosis. This is the assignment title in very first time. This will be at the top sheet. You will write up on them. And first work, it is asking for their bivalent creating phase to discuss on their figure. Okay, who are in the Zoom? I think some of this is the assignment title. Now you can see on them discussion of different stage of Prophase 1 of meiosis. And bivalent creating stage, you know about the three step you will go through on them. First one is the bivalent creating stage. You will write on them bivalent creating stage in bracket is the zygotin. So you will go through some discussion with figure, some of the features also with them. We will draw a figure on them. Very first time the layer of the shape you will go up on them. Just by your own way you will go through this one, and also draw a big nucleus inside it. Okay, in them this way, just up this one. And you know that in the leptotin where they are not arranged properly, but here we will ensure of arranging some of the figure, this one, and also draw like this way, and homologous chromosome to be set up in different way. Just draw up in the copy. We will draw like this way, and since it is a plant cell, outer cell wall, we will, and below the figure to mention the figure zygotin. zygote in them, and write up some features. Features. What are the features belonging to the zygote? We will draw up in them. Starting part on there, Ayaz Sami, Alaikum Salam, Afroz Soha, Mehrun joining in the live video. Draw up. We will try to solve the biology first paper assignment today. It is very easy because I taught you in previous time also, but some features to go through in them. Okay. Homologous chromosome, first one. We will write chromosome attract each other, homologous chromosome attract each other to make a pair, okay. Each pair of homologous chromosome each pair of homologous chromosome is known as bivalent. Homologous chromosome known as bivalent, and then bivalent. The bivalent in bracket you can write homologous pair. The pair is called bivalent. You can mark out in them also in there. This is the bivalent. Homologous bivalent can be created. Bivalent can be created at any part of chromosome, you can write up in them, and then also you will write up that the process of creating bivalent, the process of creating bivalent, the process of creating bivalent is called synapsis. The process of creating bivalent is called synapsis. Okay. And up to then you can see in the Zoom, I think, on there, and also in the live video, the proceed on. This process is go on there specifically. The process of creating bivalent is called synapsis. And then number 5, we will write up something here. Bivalent number is half. Bivalent number is half of total chromosome. Okay. And so we can say, you can write in, so in this stage bivalent created. So in this stage bivalent created. So this is the first step in the assignment. What the asking for here. Discussion of different stage of prophase 1 meiosis. First one, bivalent creating stage of zygote, and this is the figure. In very first time you will write up on there. I mark out the bivalent where created, and homologous chromosome. Why homologous? That means similar type of chromosome. When they attract each other. In the prophase 1 we have seen the enormous and scattered here and there different sort of chromosome, but which one is similar, like the same size, same features pairing chromosome, when connect together, attract together, they make a pair in each everywhere. Here this one is also bivalent. Each pair of homologous chromosomes is known as the bivalent, and bivalent, that means the homologous pair. This pair is called the bivalent, can be created at any part of the chromosome. Anywhere they can make sure to make a binding and make sure a pair. So this one is known as the bivalent. And the process of creating bivalent, you know that the homologous chromosome pair, when it will be created here, this time the process of creating bivalent is known as the synapsis. And later on, bivalent number is half of the total number of chromosome. Likely that here 1, 2, 3, 4 chromosome are in there, and half bivalent is 1, 2. That means one pair is one bivalent. Here 2 chromosome, total 4 chromosome, 2 bivalent creating here. So bivalent number is half of the total number of chromosome. I think you understand this one, and first part is completed. Now we'll go through the second part of the assignment in there. Let's move on. I think you will collect this one. If you are not connecting here properly, then you will, after publishing the video, you will collect this one. Once again, go through the uploaded part of that video. Next one, crossing over. Crossing over. Next part, crossing over. So there is first one we know about, the definition of the crossing over. Exchanging of, at first you will give some definition. Exchanging of two non-sister chromatid parts. Non-sister chromatid, exchanging of two non-sister chromatids of homologous chromosome. Exchanging of two non-sister chromatids of homologous chromosome in the Pachytene stage. In the Pachytene stage of meiosis 1, exchanging of two non-sister chromatids of homologous chromosome in the Pachytene stage of meiosis 1 is called crossing over. Crossing over. At first you will give the definition, then you will go through the mechanism. What is the mechanism going through? Mechanisms means in which way crossing over to be done on them. Then mechanisms you will write up on them. So very first time, to figure here, I will draw up in them. This one, you will, by your own way, you can draw up two figure where by your own way you can do, but this is the basic sense of having that one. And one will be the dark. You can ensure the dark part in one side, very dark, with the pencil. Use the dark part in them, very dark. And then next stage, what will happen in them, will show up on them. Same thing, this one, and it will be the single for other half, this one. And look at here, since this part is exchanging part on them, and this one to be shared, this part from dark part go through on that part, and light part from on that part. So this one to be ensured up here, especially for sharing, where they make sure the chiasma in them, what we have seen in them. Chiasma means to exchanging part, where make sure the X shape structure on them. Chiasma. In below you will write the figure crossing over, figure mechanism of crossing over, and here I will make you understand something here. A, B, C, D, E, F, G I've given in them. Likely that part also, A, B, C, D, E, F, G. This part, these are the gene non-sister chromatic to have the gene on them. A prime, B prime, C prime, D prime, you will write up on them. E prime, F prime, G prime, you will make it, when you will draw, make it a space on them. And this one is also A prime, B prime, C prime, D prime, E prime, F prime, G prime. And that part also I will show the one-sided on them. Suppose this part, when exchanging, A prime and B prime come in here, likely that A, B, C, D, make it a space of drawing, so it will be looking great. E, F, G. And this one, look at here, A prime, B prime exchanging, C, D, E, F, G. And here A, B, C prime, D prime, E prime, F prime, and G prime. And here also A prime, B prime, C prime, D prime, E prime, F prime, and G prime. Look at here. Sir, where is the change here? You can have a question in them. Question is, suppose normally which one is not dark? A, B, C, D, E, F, G. All the gene are on there. Here non-sister chromatid, this is a one chromosome, another chromosome. Here A prime, B prime, C prime, D prime, all of them. But exchanging part, A prime, B prime come here and AB go there. This is the change. This is the crossing over. After then, the gene change from the one non-sister chromatid to another non-sister chromatid. So mechanism is the 3-step here. I will make you sure here at first. You can write up here, this one is the non-sister chromatid, chromatids, and these two, any single two is the sister chromatid, and where exchanging part you can write up on them. Okay, two non-sister chromatid, two non-sister chromatids break down at the same places, by enzyme. Enzyme is there, endonuclease enzyme. First step this one, second step is on them. Then after the part of one chromosome, again bind with part of another chromosome, by ligase enzyme. You will write up, I will make you understand. Number third, that one. In this stage you can show chiasmata in bracket X created Okay, and number third stage, terminalization. Exchanging of chromatid, exchanging of chromatids end up by terminalization. Look at here. Since I have draw up here first one, the mechanism. At first you will draw the figure, these two figure, and we already give the definition: exchanging of two non-sister chromatids of homologous chromosome, same types of chromosome, which one can create the bivalent homologous chromosome. When in the packeting stage they can exchange their part, that is called the crossing over. And mechanism, three mechanism, I will draw the figure and mechanism. Two non-sister chromatid, this one is a chromosome, the chromosome, and two non-sister chromatid means one chromosome, one chromatid, another chromosome, another chromatid is the non-sister chromatid. And two non-sister chromatid break down at the same stage, where they will need to have the crossing over on that part. They will break down at the same places, but they are working some enzyme. It is endonuclease enzyme. With the endonuclease enzyme at the same place, they are breaking down. And the part of the one chromosome, so part of the one chromosome exchanging or binding with another part of the chromosome, and form the chiasmata X shape. This look like X. It look like X like. So chiasma form after then by ligase enzyme, and on that time exchanging part is completed when terminalization gone. And look at here, I tell you A, B, C, D is the one part, and A prime, B prime, C prime another part. So since their terminalization done, chiasmata formation, exchanging of chromatid in them, so A, B part exchanging with the here, and A prime, B prime part come here. So A prime, B prime dark part, these are the dark chromosome, this is a light chromosome. So dark part come here, this part A prime, B prime gene come here, and AB go there on them, the light part. So this is the crossing over done, and this is the mechanism. This way the crossing over done. If you write by drawing figure with these three mechanism step, it will be better on them. Now the importance of crossing over. This is the second part not yet completed. Some of the importance also have in them, so you will write up after then you will go through. I don't erase the figure. You will write below after then, after crossing over, when you complete to write up here, then you can below importance you will ensure up in them. Importance. So some of the importance also have in them. Genetic change, two non-sister chromatid on them, two non-sister chromatids, exchange, non-sister chromatid exchange. So genetic change to observe, and then we will go through characteristics also be changed in here, specifically characteristics, genetic, as a result of genetic change, as a result of genetic change, characteristics to be changed also, characteristics to change. Then after diversity created, biodiversity. I just write short form, you can write some elaboration in them. Biodiversity, observe, adaptive feature to grow, adaptive feature to grow. Then after new species to create, new species to create, genetic recombination, then reproduction, significance in reproduction, then we can write up here genetic mapping, then identification of gene in chromosome, and finally research. I have write the keywords, some of the key features of the importance. You can write up this one accordingly to go through detail about them. First one, it is to say two non-sister chromatid since exchange, so genetic change to be observed. Genetic change, of course, non-sister chromatid, the new feature can be created on them. As a result, genetic change, as a result of genetic change, characteristics to change. So some characters share from here to there, so new character can observe on them and characteristics to change. Biodiversity, since that crossing over on there, and biodiversity means variation of the life, we can observe on them. And then adaptive features to grow. Suppose this chromosome bearing animal or plant having less adaptive quality, but when exchanging or crossing over on them, so adaptive quality to observe. Adaptive means they have to adaptation with the environment if it is very hardship on them. And then new species to create. New species means since they crossing over done on them, so new species to create on there. Genetic recombination means gene to recombinant from here to there. Look at here, A, B, C, D, A prime, B prime, C prime, D prime. So A, B on there, and this A, B to A prime, B prime to exchanging on them. So genetic change, genetic recombination, gene here A prime, A, B, C, D, and A prime, B prime, C, D. A prime, B prime, C, D. So genetic change, gene to recombination. Here the setup ABCD, A prime, B prime, C prime. So A prime, B prime to exchange on them. And then after very last one, significance in reproduction. So reproduction significance, when we will find out that during reproduction, the crossing over is very important role. New feature to create since it is important. Genetic mapping, so we can find out the mapping on gene and genetic change on them. So genetic mapping to be counted on them. Genetic mapping means, you know, what type of feature we can make sure the crossing over, and what type of new feature to be observed on them. This is the genetic mapping. Identification of gene in chromosome, where the gene located in locus, where is the locus, we can easily find out by them. And research means always crossing over type very important topics can also the part of the research. So this one is the importance of the crossing over. Second part. And third part is last two stage of the meiosis 1, prophase 1. So we will make sure the difference. So look up on them. The last two stage of them for having a difference chart. Difference means the difference you can write up on them, headline accordingly, and make sure the diplotin and diakinesis. I want to check out just a minute, since in my same device, prophase 1 last two stage difference. Okay, So last two stage difference, we will draw a chart in like this way, like this way. So some features in the left side, features, first one, and then here last two stage is the diplotin and diakinesis. So it is needed to draw the figure, but I will draw the figure in very last time. Something to make sure that where the diplotin can occur, ensure of them. Then we will write chromosomal destruction, chromosomal repulsion. I will write up something, you can add with them also. Chromosomal repulsion, in case of their observed here, and here you can write up in them. In the diakinesis, chromosomal repulsion is not counted, not applicable. That means the stage is completed on them, not applicable. And then second stage, second point we can write up on them. Loop creation, centromere of two chromosomes, centromere of two chromosomes, repulses. So in between them, repulses. So in between them, loop created. But in the diakinesis, at the ending stage, loop is vanished, or you can write up in them, or terminate at age. This one, number third. Nucleolus and nuclear envelope. Envelope observed, and here disappear at the last stage. In this way we can go through some features. Number 4, terminalization, division of chromatid. If the though I am very ending stage, division of chromatid, go through terminalization, and here no division of chromatid, if the zoom is end up, we will go through the live. Though I am very ending moment, no division of chromatid found. Number 4 I have written, and number 5 I have written in very short way. You can go through detail about them. Number 5, exchanging part of result of chromatids start exchanging in here, and you can write up in them fully exchanged. So chromosomal repulsion observed. Repulsion means chromosomal repulsion is created and not applicable in case of the kinesis. Loop creation, centromere of the two chromosome repulses, so in between them loop created, and at the ending stage loop is vanished and terminated at the edges. Nucleolus and nuclear envelope is observed in the diplotin and disappear at the last stage. Division of chromatids go through terminalization. Chromatid is not divided on there, divided, and ending part terminalization when go on there, thereafter not, but here no division of the chromatid is found on them. Exchanging part start exchanging by terminalization and fully exchanged, and finally you will draw a figure. I just draw the figure, last one. I think you understand these points, and my link will be cut off within a short time, so I am in hurry. Number 6, you will write up the figure on there, and we can go through on them this way, this time, this way. Just very short way I try to make you understand to draw the figure. This one, this way, that one, this way, and one part will be the dark, and another part will be the light. You can make sure that one, and here also one part will be the dark and another part will be the light. This is the in below figure diplotin you will write on them, figure diplotin, and last one is the similar way, the diakinesis. I'm very quickly and hurriedly do this one, so you will do the figure properly. Okay, so this one, this way we will draw the figure in them, that one, and look at here, this one, this way, in this way, and look at here the exchanging part. What I say, this is the exchanging part, since it is the dark part. That means the crossing over what we have seen in the diakinesis, the result we found, whatever I have written in the last part. And here only one part, upper part I show in there. So this one can be observed in them, likely this way. So figure you will write the diakinesis, you can mark out some point, that one, and it is the ending part. So nuclear envelope and nucleolus is not prominently observed in there. Nuclear envelope is going through, disappeared, observed. Exchanging of chromatid, start exchanging but not yet exchange, and finally we observe there. You can see in them the observing that the exchanging part are counting on them. So this is how I give you a total guideline. You can add something from your knowledge and from the help of the book. I hope so. All of you understood the assignment of biology first paper today, what we discussed for, and now write up properly. Thank you very much everyone, and stay safe, stay sound. We'll meet once again in the next class. Allah Hafiz.